The members of the Department of Biochemistry and Molecular Biology have expertise in diverse research areas. Our work supports the research requirements for PhD, MD, MPhil, and for the oldest running 2-year MSc course in the Faculty of Medicine. Our projects have collaborative components with national organizations and some with international organizations. Environmental pollution identified within the Sustainable Development Goals affects human health, and developing methods to deal with the increase in plastics is particularly important. We isolate and investigate microorganisms, including bacterial fungal mixed communities, that are efficient in the degradation of petroleum hydrocarbons such as crude oil and polythene. We use gas chromatography mass spectrometry to analyze degradation capacity and investigate genes involved in alkane degradation in these organisms using quantitative real-time PCR and computational approaches such as protein structure prediction and molecular docking. Through successive technical cooperation grants from the International Atomic Energy Agency and international collaboration, the department has developed its capacity in stable isotopic methodology to study body composition and energy expenditure. We use deuterium-labeled water to determine body composition. Stable isotopic estimation of fat mass and fat-free mass gives us a better understanding of body fat compared with proxy measures. Deuterium is measured by Fourier transform infrared spectroscopy. We study energy expenditure using doubly labeled water and resting metabolic rate through indirect calorimetry. These gold standard methods have allowed us to develop and validate field tools for body composition, resting metabolic rate, and energy expenditure. Intervention trials include a controlled clinical trial to improve physical activity among young children through the national education system. We also explore interactions between physical activity, diet, and disease through lifestyle patterning studies. Our team are collaborating members of the Global Dietary Database, and we take an active role in nutrition advocacy at national, regional, and international level, working with the WHO and the FAO. Maternal nutrition is crucial for optimum health of both the mother and her baby. Our work aims to understand the function, metabolism, and regulation of nutrients, particularly folate, iron, and vitamin A, as well as lipids in adipose tissue during pregnancy. In studying the role of hematological indices in predicting early iron deficiency among pregnant women, we aim to provide evidence for clinical practice. We take a multidisciplinary approach to explore novel delivery systems using nanoparticles and nanohybrids, looking at increasing the bioavailability of micronutrients such as ferrous ascorbate and folic acid. We also investigate novel ways of minimizing post-harvest nutrient loss in fruits and vegetables. This collaborative work is funded by the Third World Academy of Sciences. Studying the food environment, we have evaluated the extent of commercial complementary foods in Sri Lanka as part of a regional study, and we study television marketing of unhealthy food and drink to children within a 9-country study funded by IDRC. Our department has established a cell culture and stem cell laboratory. The first US patent in the University of Colombo was received through collaborative work on isolation of a compound with anti-cancer properties from mushrooms. We are currently studying the effects of external substances on gene regulation using cancer cell lines as in vitro models, working towards new therapies.